I would also like to start thanking the Max Planck Foundation and the IMP for this great honor, and I'm really grateful for this recognition of my PhD work. And I'm also really excited that this gives me the opportunity to talk to you about what I did for the past couple of years. And so in my PhD, I was interested in an oocyte-specific cytoplasmic structure called cytoplasmic lattices. And in the next 10 minutes or so, I'll take you through how we came to understand how cytoplasmic lattices are important for the accumulation and storage of maternally deposited proteins that are needed for the early embryo. Broadly speaking, I'm interested in how the egg cell prepares for and contributes to the development of a healthy embryo and ultimately the birth of a healthy new individual. And normally, when we think about the maternal contribution of the developing embryo, we often think about the genetic contribution, because we, of course, know that the error-free genetic contribution of the egg to the embryo is essential for proper development. But while chromosome segregation and mechanisms of aneuploidy has been an active field of research in oocyte biology, a lot less is known about the cytoplasmic contribution from the egg to the early embryo. And this is important because in the cytoplasm are stored a lot of the components that the early embryo needs, because these first stages of embryogenesis are transcriptionally silent. And so because I was interested in how the egg cell cytoplasm contributes to early development, I thought, well, let's start with just looking at what is actually inside the cell cytoplasm. So here you see an electron micrograph of a mouse oocyte. And in the electron micrograph, you can see structures that you would expect to see in the cytoplasm of a cell, such as the mitochondria and the plasmatic reticulum. But in mammalian oocyte cytoplasm, there's one structure that is extremely abundant, and this structure is called cytoplasmic lattices. And you can see them as these sort of striated filamentous structures here that maybe you can appreciate better if we zoom in a bit. And these structures, they take up about 10% of the oocyte cytoplasmic volume, and they were discovered in the 1960s with electron microscopy, but has since then puzzled scientists because we didn't really know what they were made of, nor what their function is. What we did know, however, and what makes cytoplasmic lattices a really important topic of research, is that in embryos from both mice and humans that didn't have cytoplasmic lattices, their progression would be halted or arrested as early as the 2-cell stage. And so basically, we have this extremely abundant structure in the oocyte cytoplasm that is somehow essential for early mammalian life, yet we don't really know much about it. And so my PhD started with an electron micrograph, essentially, and the question, what are cytoplasmic lattices? What is this structure that is so abundant, filamentous, that is so important for early life? And can we, by studying this structure, learn something about the maternal cytoplasmic contribution to early development? When I started this project, we didn't really know much about cytoplasmic lattices, but one thing we did know was that cytoplasmic lattices have been linked to oocyte-specific proteins called PADI6, and proteins of the subcortical maternal complex, or the SEMC for short. And it was found that when PADI6 or the SEMC proteins are knocked out, or the genes encoding these proteins are knocked out, cytoplasmic lattices no longer form in the oocyte cytoplasm. And PADI6 and the SEMC proteins have been shown to localize to the oocyte periphery, or the subcortex, as the name suggests. But I said that cytoplasmic lattices are everywhere in the cytoplasm. So the link between these two structures was not really clear. But nevertheless, these provided me with a couple of gene names or protein names that could start this sort of investigation of the cytoplasmic lattices. And so I decided that in my quest to understand what the cytoplasmic lattices are doing, I first needed to establish the link between PADI6, the SEMC, and the cytoplasmic lattices, hoping that this would give me some direction into the question of what their function is in the oocyte and the early embryo. So to answer this first question, I first needed a way of reliably visualize and label these proteins inside the cell. Now, I said that PADI6 and the SEMC have been shown to localize to the oocyte periphery, but this was sort of debated in the field with a lot of different research groups finding a lot of different subcellular localizations of these proteins. Common to all of them, however, was their use of indirect immunofluorescence or antibodies. So to kind of circumvent the use of antibodies, we made a knock-in mouse line in which we inserted a halotag sequence into the endogenous locus of PADI6, one of the proteins important for cytoplasmic lattice formation. And halotag can be made fluorescent by incubating the cells with a fluorescent ligand of the halotag protein. So for the purpose of this talk, I will talk about PADI6, but what we found for PADI6 also held true for the SEMC proteins. And as you can appreciate from this image here, this is a mouse oocyte expressing the fluorescent version of PADI6. And as you can see, PADI6 was not restricted to the oocyte subcortex, rather it was distributing throughout the cell. And interestingly, we found that the previously reported subcortical localization of these proteins were actually due to insufficient secondary antibody penetration, which can be a problem in large cells such as oocyte or larger tissue sections, for instance. And interestingly, looking closer, we found that PADI6 as well as the SCMC protein was organizing sort of short filamentous-like structures in the oocyte cytoplasm. So now we had a group of proteins that were distributing throughout the cell, that were forming short filaments, and were important somehow for cytoplasmic lattice formation. So the next question was then, are PADI6, the SCMC proteins, actually colocalizing with the cytoplasmic lattices? Or in other words, are they the same structure? So to answer this question, we needed now to visualize our proteins of interest within the ultrastructural context of the cells, colocalizing them with the structure that has only been seen with electron microscopy. And to tackle this problem, we actually used pan-expansion microscopy, a relatively newly established technique, which allows you to image your proteins of interest within the ultrastructural context of the cell using conventional light microscopy. So expansion microscopy is based on the principle that you can achieve super resolution or high resolution by physically expanding your cell by embedding it in a swellable hydrogel. And pan-expansion microscopy is the latest addition to the expansion microscopy toolbox, using a fluorescently tagged NHS ester that will bind to primary amines of your proteins in your sample, creating a fluorescent tag of all the proteins within your cell. And this allows us to get a sort of electron micrograph-like image of an oocyte cytoplasm using light microscopy. And importantly, this allowed us to visualize the cytoplasmic lattices within the oocyte using normal conventional confocal microscopy. And so the cool thing about expansion microscopy and pan-expansion microscopy is that not only does it give you kind of a whole holistic view of the whole cell cytoplasm, you can also combine it with antibody labeling. And so if we go through this C-stack of a mouse oocyte and overlay it with the signal of PADI6, for instance, we see perfect colocalization between the two structures, indicating that the cytoplasmic lattices are in fact made up of PADI6 and also the SCMC proteins. And this is really important information because now we know that when we're knocking out the genes encoding PADI6 and the SCMC, we're actually disrupting the building blocks of the cytoplasmic lattices and not some upstream regulator. And this now gives us some idea of how to answer these questions of the function of these structures. So because we now have a way of visualizing cytoplasmic lattices using PADI6 tagged mice, for instance, we can look at cytoplasmic lattices throughout the pre-implantation embryogenesis. And what we see is that the cytoplasmic lattices actually persist quite long in the embryo and disappear just before the embryo implants at the blastocyst stage. But I told you that embryos without cytoplasmic lattices will stop dividing already at the 2-cell stage. So we were wondering whether the cytoplasmic lattices might be harboring some of these maternally deposited factors that are deposited in the oocyte and helps drive embryogenesis through these early stages while the embryo is dependent on factors deposited in the mother. And so we were wondering whether the cytoplasmic lattices might be storing maternally loaded proteins. And to answer this question, we compared the oocytes with and without cytoplasmic lattices using mass spectrometry. And if our hypothesis were correct, we would expect that upon depletion of cytoplasmic lattices, we would see a depletion of maternally loaded proteins that are important, maybe not in the oocyte, but later on during embryogenesis. And in fact, this is exactly what we see. When we knock out PADI6 and components of the subcortical maternal complex, we actually see a lot of proteins less abundant compared to the wild-type control. And importantly, we found that there was no change on the mRNA level of these differentially abundant proteins, meaning that their less abundance were not due to a downregulation of their respective transcripts. And more excitingly, we found that when we looked at the identity of these proteins, they were important for a lot of cellular processes that are important just in the very first stages of embryogenesis, allowing this transition from maternal to zygotic control of development, such as the reprogramming of the epigenome, protein-protein clearance, and even formation of microtubules. But now the next question was, how is the cytoplasmic lattices actually regulating the abundance of these proteins? And one idea we had was that these proteins might actually be localizing or sort of the cytoplasmic lattices might be serving as sort of a scaffold to allow the accumulation and sequestration of these proteins. And to test this hypothesis, we could go back to our expansion microscopy setup, which allows us to efficiently probe the different mass spec identified candidates for co-localization on the lattices using PADI6 as a marker. And so we checked a lot of the different depleted proteins involved in different cellular processes important in the early embryo and found that proteins involved in chromatin modification and DNA methylation maintenance, such as UTRF1, proteins involved in protein-protein turnover, such as SCIP1, and even a non-filamentous pool of tubulin was localizing to the cytoplasmic lattices in wild-type mammalian eggs. And this now led us to propose that the cytoplasmic lattices composed of PADI6 and SCMC proteins is serving as a maternal protein storage compartment, allowing the faithful development of the early embryo. And in contrast, loss of cytoplasmic lattices leads to loss of these maternally deposited proteins, causing the embryo to arrest at these very early stages. And excitingly and importantly, these results now also allow us to explain some of the other phenotypes that are seen in women with SEMC or PADI6 mutations that could previously not be explained by PADI6 and SEMC mutations alone, such as the formation of molar pregnancies or, in rare cases, birth of children with imprinting disorders. And these two phenotypes are both hallmarked by aberrant DNA methylation patterns, which can now be explained by loss of these proteins regulating these when the cytoplasmic lattices are lost. And so to summarize, in my PhD, we answered two long-standing questions in oocyte biology, namely, what is the function and the composition of cytoplasmic lattices, and how does the oocyte store proteins that are needed for early embryogenesis? And so we were kind of imagining the cytoplasmic lattices as this sort of cellular storage compartment where cytoplasmic lattices act as shelves that allow the storage and the synthesis of proteins that are stored away until they are needed after fertilization, as beautifully illustrated in this illustration. And so with that, I'd like to thank you for your attention. I would like to say a specific thank you to my amazing supervisor, Melina, who's been absolutely fantastic to work with, as well as the entire SHU lab. And also a specific thank you to the collaborators on the mass spec side, Luisa, Henning, Yehor, Ina, and Juliana. And also to our collaborators on the cryo-electron tomography, which I didn't have time to go into today, but we had an amazing collaboration with Ruben Fernandez Bisnadiego's group, where we used cryo-electron tomography to try to elucidate the molecular organization of the lattices inside the oocyte cytoplasm. So thank you.